Okay, welcome everyone. It's 4 o'clock. My name is Sharon Wolf from the Weizmann Institute, and I'm here to chair this 38th Structure Meets Function webinar, sponsored by Instruct. So as you can see on the slide, that Instruct, what is Instruct? It's Instruct-ERIC is a pan-European research infrastructure focused on advancing structural biology. It provides access to cutting-edge technologies, expertise, and training to researchers worldwide, with a particular emphasis on fostering innovation in biomedical sciences. There's access to researchers from member countries, which you see on this map, and they can get funded for specialized services ranging from sample preparation to 3D structural analysis. And Instruct-ERIC offers training by workshops, internships, and training programs in emerging structural biology techniques. So I'll stop sharing my screen so that I can introduce our first speaker. We will have 2 speakers. And the first one is Stefano Morasso from Trieste. Stefano received his PhD in chemistry from the University of Trieste in 2024. And he's been working in the group of protein targets for drug discovery, headed by Dr. Paola Storici at the synchrotron in Trieste. You can correct her name for me.
That was great.
Good. Exploring viral enzymes and human kinases as crucial targets. So he's currently undertaking an Instruct-ERIC funded internship at the Institute for Structural Biology in Grenoble to expand his expertise in protein production and MR. And the title of his presentation is Boosting Drug Repurposing, Allowing a Multi-Technique Approach, a case study in SARS-CoV-2 PL-Pro. So please go ahead, Stefano, and remember, keep on time.
Yes.
I will unmute myself to let you know you have a couple of minutes left.
So let me share my screen. Just a few seconds because my team application maybe it's slow. Can you see my screen?
Yes, I can see your screen. It's not in presentation mode yet.
Okay, I will start the presentation mode. Okay, so fine?
Yes, we can see you. Go right ahead.
Great. So thank you very much, Sharon, for the kind introduction. And good morning, everyone. I'm Stefano Morasso. I'm currently speaking from Grenoble at the Institute of Structural Biology. I wanted to say it in French, but it's not my first language. I'm speaking from Grenoble, but actually I'm based in Trieste. I work as a postdoc at Elettra synchrotron in the group of Paolo Storici, indeed. And as you can see, maybe I can use the pointer. Yes. So as you can see from the name of our group, we are interested and we work on proteins that are interest of medicinal chemistry projects. And indeed, today, despite the variety of targets we use to study, I'm going to talk about a viral target. So, of course, it's on SARS-CoV-2, and as many others groups, we started our research and experience with SARS-CoV-2 at the beginning of the pandemic, while we were involved in the Escalate for COVID project, which was a European-funded project headed by Dompe Farmaceutici. And the main aim of the project was to identify new inhibitors for different targets of SARS-CoV-2, because, you know, we were in an emergency situation, so we needed to speed up the research on this virus. So our group in Elettra, we joined the project and we focused our attention on the system proteases. So I am sure you know by heart the viral replication cycle of the SARS-CoV-2, but just let me remind you that the 2 system proteases are essential for the replication since they can release the active enzymes necessary indeed to go on with the replication. So they're really crucial targets for the treatment of this virus. And let me say also that we had quite a straightforward experience with the main protease, or NSP5, which gave us quite a lot of success and satisfaction, especially from a crystallography point of view. But we struggled a little bit more on the papain-like protease, or PL-Pro, and that's why I'm going to talk about this target in particular today. So the PL-Pro activity is not only interesting from a viral point of view, because of course it's essential for the viral replication, but it's also essential for the virus because it's one of the main way this virus can escape the immune system responses against the virus itself. Because it has not only a protease activity on the viral proteins, but also it shows a desigilating and deubiquitinating activity. Indeed, we can define a particular fold or shape on the structure of the PL-Pro that is called the finger, thumb, and palm, which is typical of the USP family of the ubiquitinases. And also from the co-crystal structure with the ISG15, we can define two binding sites. So one at the top of the thumb, called S2, that binds the N-terminal domain of the ISG15, and a second binding site, the S1, that is close to the catalytic triad. And it's also the main target pocket of all the inhibitors, or most of them, that came out during the research on this target. So the first repurposing and also the first to be validated as repurposing inhibitor was this GRL compound. I like to call it the single father of the PL-Pro inhibitors that bind indeed the S1 site, so competes with the ISG15 for the binding, so with the substrate. But I like to call it single father because it was the only one that generated novel molecules that, especially during last year, raised the attention on this target and also demonstrated quite high affinity and potency against this target, and also protecting from the virus itself. Indeed, they were proven to be active and effective also in vivo model, but they share, all of them, this scaffold of the GRL. So our main objective was and is still today identifying new inhibitors with different chemical scaffolds that can be considered for the drug development and so on for the treatment against the virus. So as I said, we are first of all a protein production facility. So we produce two constructs actually, the PL-Pro itself full length. But since this protease is a subdomain of a larger multi-domain protein that is the NSP3, you can see the scheme here, we decided to produce also a larger construct, including a nucleic acid binding domain at the C-terminal of the PL-Pro, that is the following domain in the sequence of the NSP3, with the aim to check if the inhibitors were still working with the same behavior against also this other construct, and also to highlight, if possible, some new druggable pocket maybe on the other domain. So we produce the protein in E. coli, and I would say as all, it happened to me to find this statement in all the congresses and all the seminars I attended about structural biology, that the quality of your sample is a key factor on the results of your structural studies. And indeed, it's what we believe also. So we care a lot about the quality control of our products and protein we produce. So not only in terms of purity, but also of homogeneity and thermal stability. For example, in this case, we highlighted the fact that this protease, both from activity point of view and stability point of view, needs a reducing environment to be safe. And at the end, we could also obtain some apoform crystal using the shorter construct, so the PL-Pro itself, let's say, which diffracted quite well. So we were ready also to move on working with the inhibitors and maybe use the apoform crystals for the soaking. So we were involved within the Escalate project with a large, medium-large, repurposing screening conducted at the Fraunhofer Institute in Hamburg, where they developed a double activity assay using a labeled peptide or the labeled natural substrate IC15. And after all the screening, they came out with a very promising cluster of compound with ortho or para-quinone-like scaffold with a range of affinity between low micromolar and low nanomolar. In particular, our hit compound was this one, that is varicin B. It's an old antibiotic, not really used anymore, but it showed a low nanomolar activity, so we were pretty impressed and also excited, and we wanted to start characterizing the binding. So I started with co-crystallization, also soaking with our pretty nice apoform crystals, but the only results I could get was precipitation. So I tried to validate the binding in a different way, starting from thermal shift assay. At first with the known inhibitor as a positive control, so the GRL compound, as you can see, it behaved as we expect from a small molecule in this kind of assay. So we had a strong positive thermal shift. So it worked, the assay. But when I tried with our heat compounds, I got the opposite, the opposite result. So all of our heats were strongly unstabilized this protein, really going into unfolding. That was also explaining what I saw in the precipitation during the crystallization trials. But that wasn't what I wanted to see. So I tried also different ways, like with limited proteolysis, but also here we had the same results. So as you can see, the combination protein plus the compound strongly destabilized the protein. So we could see a stronger digestion from the protease. So in the end, we came to the hypothesis that there was something wrong. There was a side reaction. And in particular, we started thinking that, also supported by literature results, that this compound could react with the strong reducing agents like DTT, as you can see here, in the environment of the protein. This reaction could cause some strong oxidizing factors like superoxide species that in turn react with the sensitive catalytic cysteines, like we have in our protease, causing a non-reversible oxidation and in the end, the unfolding of the protein. So let's see what happens if we don't use reducing environment in our assays. We completely lose the inhibition activity of our inhibitors. We don't see the strong destabilizing effect at the thermal shift assay. As you can see here, if we don't put anything, basically we don't have any perturbation of the protein. And as a final demonstration, our colleagues in Naples quantified and characterized the oxidation state of the catalytic cysteines in presence of both the compounds and the reducing agents. So we finally got into false positives. That's why we revised and we got back to the screening. This time we considered milder reducing environment also because I remind you that this protease needs some reducing agents to keep its stability and activity. And we finally came out with, this time, a true inhibitor, the CPI compound that is a methyl histone transferase inhibitor. Not so small, I would say, with a low, medium, low micromolar range affinity, here expressed in IC50. So this time I started the validation from the thermal shift assay before going into crystallization. And as you can see from this graph, the CPI behaves in a similar way to the GRI compound, both on the wild type and also on the catalytic cysteine mutated. So we could say that the catalytic cysteine was not involved in the binding directly with the CPI compound. From docking prediction, we highlighted indeed the share the same binding site, so the S1 site for the ISG15, the C-terminal of ISG15, but we saw at similar probability two different conformations of the CPI. In particular, the indole portion is basically at the same position, but the rest of the molecules enter the pocket in the opposite way. So now we had to validate, of course, this prediction, but also understand which one of the two conformation was the most probable. So we needed some structural data. This time as well, I tried largely with co-crystallization or with soaking with our apoform crystals, but again, we didn't get any data set useful to determine the structure. So we finally decided to change our strategy. And we got a great assist by NMR, thanks to a collaboration with the Slovenian NMR Center in Ljubljana, which hosted me for the experiment. And also they taught me a lot, and they followed me developing these experiments. And in particular, we studied the binding through saturation transfer difference NMR. That is an based on intermolecular transfer of magnetization from the molecular target to a reversible ligand. So it's based on the fact that if we saturate specific frequencies corresponding to the protons of the target, but without directly perturbating the ligand, for cross-relaxation, the saturation is transferred also to the closest protons of the ligand. In this way, we can also describe the binding epitope of the ligand in the binding pocket from a ligand point of view. As you can see in the CPA's case, the binding epitope is quite distributed along all the molecule. But to identify if the S1 site was really the binding site, we performed a competitive assay using the GRL, which structure known. So we could see not a fully but consistent displacement of the CPI once we were adding the GRI compound. So we could say that the two compounds were sharing the same binding pocket. But in the end, we also wanted to validate one of the two conformations we predicted with the docking. So for this, we changed experiments, and we did two-dimensional experiments this time, transfer noisy. It is based similarly to the SD experiment to the transfer of saturation from the macromolecular target to the small ligand. But in this case, we look at the intramolecular distances within the ligand. So our transfer noisy data match better with the second predicted pose in the S1 side. So with the pyridine moiety sitting outside the binding pocket, and with the ethyl sulfone moiety going toward the catalytic triad. In the end, we were able to identify a new inhibitor with some similar features, chemically speaking, with the GRL, but a different scaffold. We described, of course, the binding mode using different approaches of computer-aided NMR and biophysical assays, and combining all together. We tested also, but our collaborators in Cagliari tested the effectiveness in antiviral protection in a cell-based model. And as you can see, the antiviral effect, it's appreciable, but at high concentration. So we found an inhibitor, but we would say that must be optimized. Then, meantime, we were also involved in a larger screening, collaborating with the European Lead Factory. This time was not reporting screening, but they screened a quite larger compound library, and they find another cluster quite different from GRL that now we want to validate to, of course, optimize as well as the CPI. That's why we joined last year the Electron project. So you've been watching to the logo all this time in all the slides, so I think it's time to present this project now. It's an European funded project started indeed last year that has the aim of developing solid pipeline, especially at the clinical level, for the development from the identification and then the optimization of antiviral compounds, especially if we would like to develop a broad spectrum antiviral drugs in order to be prepared, of course, for future issues, because I don't want to say pandemics, but issues for sure. And so we are divided into 8 work packages based on our skills and the field of our research. We are currently in the work package 1, so drug discovery and medicinal chemistry. And at the moment, we are keep on studying the PL Pro and moving research we have years on the PL Pro, but we're also considering other targets from SARS-CoV-2, like the helicase, and now we're also open to evaluate different targets of other viruses like dengue or other.
Stefano, you're breaking up a bit, and also you have about another minute. Okay.
I'll be sure now. Thank you. So our aim is to provide a solid pipeline from the protein production, and we, as I said, we care a lot about the protein control in order to provide proteins and our results to our collaborators. And from a binding point of view, both biophysical binding and structural point of view, we are trying to enlarge, also learning from the past, our knowledge and our possibilities. That's why I applied to the Instruct-ERIC internship to explore a bit more NMR, because I would like to perform this time a protein observed NMR. So I needed to prepare isotopically labeled PL Pro. So I'm hosted here at the Institute of Structural Biology by Jérôme Bouvier and Lionel Imbert. And I think the aim of this internship in general is not only to go on with your experiment and to allow you to try experiments you cannot do at your formal lab, but also to learn something. And that's why I'm learning from the experts how to use cell-free production that is quite convenient for isotopic labeling, and also to how to deepen into this system and to how to set up it for future development in my formal lab. So at the moment, I have just preliminary results, but they are positive. So I've been working with the PL Pro and other 2 fragments. I expressed them successfully in a cell-free system with the help of Lionel Imbert. And now we are currently trying to optimize the yield and the solubility of our construct. So in conclusion, we we established a solid pipeline at Elettra to validate the compounds in drug discoveries campaigns we are involved, also learning from the past. And thanks to the opportunity that was given to me from Instruct and of course my hosting group here in Grenoble, to expand my knowledge and possibilities both from a protein production point of view, but also from the structural analysis. So let me thank all the collaborators I had the pleasure to work with, especially from my former lab in Trieste and also from my hosting lab here in Grenoble. And I would like to thank you all for the attention.
Thank you very much, Stefano. We have a few questions in the chat. I don't see the Q&A, but I'm told by John that he'll forward anything from the Q&A. And they're all from Filippo Perrigo. He says, ciao, Stefano. And he asks about, first of all, nano differential scanning fluorimetry.
Yes.
That maybe, did you try concentrations that were high enough? Because it seems to him that using low concentrations such as 0.2 mg per mil would not give a nice signal.
Well,
hold on. I think, Celia, don't share quite yet your screen, please.
No, it's still me, I suppose.
Somebody's sharing. Okay, there we go.
Yeah, it's me. I can stop the sharing, actually.
Yes. So can you answer that question? He asked if you tried nano DSF.
Yes, I tried also to exceed more the ligand protein ratio in favor of the ligand, of course. And with our, let's say, true inhibitors, we had quite nice thermal shift with them. Also at quite low concentration, but I mean, with 10-fold excess, we could appreciate quite a nice thermal shift. So, I mean, we were quite happy with that result as a first qualitative validation. So yeah, we tried as well also for the co-crystallization, of course, and also for the soaking. I exceeded much more, but without any good results up to now, but we're still working on that.
Okay, and he also has a question about the crystallization solution that you use. He finds that PO4-based solutions work well, but in the case of some compounds, soaking dissolves the crystals. Do you have any advice?
I have some experience in it, actually. It really depends, I would say, on the crystallization conditions. For me, for example, in my case, my apoform crystals grew in high salt concentration conditions that is not really suitable for such insoluble small molecules. So that's not really helpful, but if you can manage to find more polymers based that can help the co-solution of the compounds in the crystallization drop, maybe might help a bit the soaking and also, of course, co-crystallization if you try to co-crystallize. But of course, depends then on the protein and the combination of protein and your compounds.
Yeah,
so
of course.
A lot.
I have another question here from Arnaud Potzerman. Did you evaluate the effect of DNA in the case of the long construct, which, if I understand, comprises a DNA binding domain?
Yeah, it's true. Thank you for the question. I didn't actually. I actually worked quite a lot to clean out the DNA from that construct while I was producing it, because indeed the nucleic acid binding domain is proved to bind especially RNA. So for the viral, the hypothesis is that it's driving the RNA in and out the viral vesicles, because the NSP3 together with the NSP4 makes a pore in the viral vesicle, and the nucleic acid binding domain is really at the entrance of the pore. Oh, I see I'm frozen. I'm sorry. My camera is not working.
No, it hasn't been working. Just leave it alone.
Sorry.
Yeah, it is. Yeah, we have to switch to speakers, but just finish your answer then, and we'll.
Yeah. So I didn't test it, but it's actually quite a nice point, because it's something that is not explored up to now, I would say, at least from literature, I haven't found anything about that. But what we know, maybe more from the experience of SARS-CoV-1 nucleic acid binding domain, it's meant to bind RNA. That's what I can say.
Okay, thank you. We've run out of time. We have to move on to our next speaker. Stefano, thank you very much for your very clear and well-presented lecture. We now have Celia Romao speaking. Celia is a researcher at ITQB in Lisbon. Her PhD is also from the ITQB, and she postdoced at the ESRF in Grenoble. Her main research focus is on elucidating the molecular mechanisms that underpin radiation resistance, with a specific emphasis on Deinococcus radiodurans, which is recognized as the most radiation-resistant organism known to date. And since the start of the recent pandemic, she's also been deeply engaged in SARS-CoV-2 research, particularly studying NSP1 and its interaction with metals. In fact, Celia and I are good friends because we worked together when we applied for and received an ERC widening grant called Impact. And here's my Impact mug, which went from 2019 to 2023. And Celia has used Instruct Access to obtain data, EDX data on right here at the Weizmann Institute, and she's going to be showing some of that data today. So Celia, please share your screen, and I will wave to you about 5 minutes before, like at 5 minutes before, so that we have time for questions.
Yeah, okay. So I guess you are seeing my screen, right? And the sound is okay, right? Yes? Sharon?
Yeah, perfect.
Okay, good. So, first I would like to thank this fantastic opportunity to present our work. And before I start to present our work, I want to give to acknowledge two projects that were really important for this project. The Impact, which Sharon just mentioned, which was a twinning project that aimed to build our cryo-EM knowledge here at ITQB. And Instruct. Instruct, through access proposals, allow us to access the Israel Center, so in the Weizmann Institute, and the data that I'm going to show was collected within this framework, and it's amazing as you will see it. So our focus and what I'm going to present is the electron-dense granules, so as store compartments for metals and how they respond to a stress stimulus. So we have been studying Deinococcus radiodurans, and this is a gram-positive bacteria, so it's a coccus, and it is widely known for its resistance to radiation, to different types of radiation, and also desiccation. So the question that has been ongoing since the beginning of the discovery of this organism is why it is so resistant. And in fact, this organism contains different strategies to deal with the radicals that are formed due to exposure. So, for instance, DNA has genomic redundancy. It was proposed at a certain time that, in fact, the toroidal structure of the DNA was also important. It has a very high efficient DNA repair systems. The membrane composition is composed of 8 layers and is quite unique. And in this layer, it is present a carotenoid called diauxanthin. Here we see cells that were exposed to increasing doses of X-ray, and as you can see, the initial, so the bacteria is kind of pink, but when they are exposed to stress, especially radiation, they become more like yellow. So indicated that in fact diauxanthin is scavenge the radicals that are formed during the stress. Also, and these organisms contain a pool of manganese that forms complex with small molecules, and one of the molecules is phosphate. And these complexes are supposed to have antioxidant properties that protect the proteome against the radicals that are formed during exposure. So we have been interested to understand how manganese is regulated, where and how it is stored. Is there a free manganese pool inside the cell, or are they in fact stored in the compartment, in the specific compartment? So for you to know and for better understand the image that I'm going to show you, this is just a TEM section that we of one of our studies, and this is a tetrad, so it's composed of 4 cells, and each of these is a cell. Okay, so we wanted to know how the bacteria respond to stress. So we started to do some growth curves under control conditions, and which is here represented in black. And when the cells are subjected to stress, and here we are inducing oxidative stress by the addition of methyl violet chain that generates a superoxide ion in intercellular. And what we can see is the green line is that the cells enter into a lag phase, and then later they recover, and they recover up to the same similar levels as in the control. Quite interesting. If we pre-treat the cells before we had the stress, the cells do not feel any stress at all. So indicate that in fact manganese has some protection mechanism to the cells. So we wanted to know under these conditions what happens to the metals, where are the metals localized. So we have done X-ray fluorescence imaging at SRF using the nano imaging beamline, so ID16A, and here it is presented. So by the first image is the phase contracts, and these images correspond to the different elements that we detect. So comparing the manganese, phosphorus, and calcium, what we can see is that they co-localize in the same place, and which are a specific region, and this is consistent with a region called electron-dense granules. In fact, by looking to the image that I've shown you before, this TEM section, we see that these dark regions correspond also to these electron-dense granules. So the question is, what happens to the cells that were induced, that were subjected to oxidative stress? So when you look to the cells under these conditions, what we see is that we still see some co-localization, but most of there are still parts that are localized in the cytosol. So this indicates that in fact, during this condition, there's a dislocalization of the different metals from the electron-dense granules to the cytosol. Quite interestingly, and I'm not going to focus on this, but it's quite interesting that under control conditions, iron is located in the septum region, while in the oxidative stress is no longer, it's basically spread all over the cytosol. So, but as I mentioned before, manganese has a protective effect, and we wanted to know how the metals were localized in the conditions where we pre-treat the cells with manganese. So by X-ray fluorescence image, we could see that in fact, so each of these is a cell, and there are cells that contain a huge amount of manganese co-localized with phosphorus and calcium, and in fact, in these conditions, we see also potassium and copper. So there were also possible to see the other elements, for instance, iron, chloride, copper, and zinc, and this was excluded from these electron-dense granules. So very interesting, like you can see sulfur is completely and chloride is completely excluded from these regions, and this indicates that based on our data, that the only contraria that is present in these regions are phosphorus, the only anion. So we wanted to know which form is this phosphorus. So we using NMR, what we have detected is the level of polyphosphates, and their cells contain a huge amount of polyphosphate, both in exponential and stationary phase. So the presence and the high amount of phosphorus that we detect by X-ray fluorescence imaging of phosphorus is mostly probably in the form of polyphosphate, and this corroborates data from other authors. So we decided to continue the study, but in this case, we tested two different media. So Deinococcus is commonly grown in what we call the TGY medium that contains tryptophan, yeast extract, and glucose, and also M53 that besides the three components, three basic components, it has 0.5% of salt. So by microscopy, what we saw was that in TGY, the cells are mostly organized to 2 to 4 cocci, and in M53, they form these multicellular clusters in which they are more like between 8 and 60 cocci. So the next question was that, are these multicellular clusters, are they related with the resistance to stress? So to address this, we did first we analyzed the exposure to UVC radiation. So using the typical spot assay in which we serial dilute the cells, and then by we expose the cells by increasing doses of UVC, and after analyzing the data, taking the survival fraction, we can see that in fact cells grown in M53, which is the one that has 0.5% of salt, is more resistant than the TGY. So obviously, the next question was that, does it happen again also when cells are subjected to oxidative stress? And we did that using the methylvilogen that I mentioned before, generates superoxide anion. And what we did was that we plate the cells, and we have used an adapted method for this diffusion, where we put different concentrations of methylvilogen, and what we measure is inhibition halo. And as you can see, under this condition, the bacteria is resistant up to 10 millimolar of methylvilogen. So when we did the same experiment using M53, we had to use higher concentrations of methylvilogen to have the similar inhibition halos. In fact, we are comparing that in TGY comparing to M53, we use around 5 times more concentrations of methylvilogen in order to produce the same effect. So this indicates that, again, the cells grown in M53, so in the presence of salt, are more resistant than in TGY. Okay, so we wanted to go deeper in our analysis, and for that we took advantage of our impact project and also Instruct through access proposals to access the microscope unit at the Weizmann Institute in collaboration with Sharon and Michael Elbon. So what we did was to analyze the cells by cryostem and coupled with DDX in the cells growing in TGY and M53. So what we did is we cultured the cells, we wash and resuspend, we vitrify the whole cells, and then we collect tomograms at barren tills. So using the STEM detectors that and the setup that they have in the Weizmann Institute. So these were the first experiment that we did. So the cells grown in M53, and we use PBS to wash the cells because it was kind of standard to use PBS. And as you can see, so this dark region corresponds to the electron-dense granules, and we can observe phosphorus in these granules as we saw before. But for instance, due to technical reasons, we could not detect magnesium in the granules using X-ray fluorescence imaging. And with cryostem, we could see magnesium quite well that is also stored in these granules. Surprisingly, in what we are seeing here, we are seeing these are 2 cells and these are 2 cells. And in 2 of them, we see potassium. It's quite well detected. And in the case of the other cells, we could see that it contains potassium. So this suggests that, in fact, granules within the same cell culture can store different cations. And this maybe is related to a certain degree of organization, specialization, and in fact, resource allocation of the different metals. Analyzing another cells in the same condition, we can detect all these different metals, but also calcium was detected. So, and this agrees also with the previous data that we have collected. And when we analyze the cells that were grown in PGY, so that we didn't have any salt in this medium, and cells washed in PBS, phosphor, magnesium, and were detected, but surprisingly, we also detect in a lesser extent sodium, but we were question the origin of this sodium because we didn't have any sodium in the grow culture. But then we remember that, in fact, we are using PBS, and PBS contains a lot of sodium. So obviously, the question, what we did next was to prepare the cells in which we didn't wash them with PBS. So we have used cells both from PGY and M53. And when we compare the data from the 2 conditions, we can see that in all of them, we have a higher number of granules in the M53 conditions, but all of them contains phosphor, magnesium, and potassium, but no sodium. So in fact, the PBS affects the electron-dense granules elemental composition, and this goes behind is that the environment is definitely crucial to change the composition of these electron-dense granules. So taking advantage of the data that we have collected, we could perform a cryotomography dual accent, and here is the video where we can see different within the cell cluster. We can see different granules. We can see membrane vesicles, but this has to be for a later talk. And also, like, you can see these type of granules that are completely different from from others that were shown. And also DNA was also observed. And we see these granules that are basically uniform. And quite interesting, there within these clusters, there are cells in a different diffusion state. So this one was not divided. And what we see here is as a dark region, we see that these are metals or the granules that is shared between 2 cells, which is not fully divided. So based on these results, we could identify 3 types of granules, what we call the type 1 uniform spherical dense granules, a type 2, which are spherical granules with hole inside, which are here represented, and type 3 is amorphous granules, and here this one is the one that is moving from one cell to the other. So obviously, our question was that, and we measured then the EDX to make sure that that we were seeing the same elemental composition, and in fact, there is no difference between the composition of elements between the different granules. So we don't know yet the difference and what makes these granules look different. So we are still trying to understand what's going on with these different types of granules. So also we wanted to know the response to stress. So we took advantage of our another experiment that we were doing at SRF on X-ray fluorescence image on ID16B, in which we were analyzing a cluster of cells, and here the signal for the phosphorus and manganese and calcium. And what we did was to take advantage of the X-ray beam just to irradiate in a very specific point the cells with a boost of damage, of radiation in this case. And what we did after was to measure and to image this cluster of cells. And what we can see is that there is a distribution of elements, not only in this cell that was irradiated, with all the other cells that were part of this cluster. So this indicates that, in fact, not only the cells was affected, but all the other cells sense the stress, and they diffuse the metals and phosphor from the storage compartment throughout the cytosol. So this is still an ongoing project, but our take-home message was that these electron-dense granules act as reservoirs of different elements: manganese, calcium, potassium, sodium. Phosphorus is the only counter-ion that we have observed in these regions. Phosphorus is probably mostly in the form of polyphosphate. And in the presence of high content of phosphorus, there's a mobilization of the different cations in the electron-dense granules. And what we have observed is a new organization in terms of metals, like cells that are selected for potassium or sodium. So this raises a question: is there any bacterial communication for metal content? And what we are seeing is that this localization of elements are basically triggered by stress exposure. And I'm really grateful for doing this work in collaboration with the team in the Weizmann, because they really pushed the limits of the cell imaging in cryostem, because we didn't do any feed milling. So this is really the data from cryostem. This is amazing to get information in terms of the different polyphosphate granules morphology. So we will continue to understand these granules, and if they are spread in other bacteria, especially if they have a different and similar function. So I want to finalize by acknowledge all the team, especially my PhD student, Andrea Gouveia, and a special thank to the Weizmann team that was fundamental on all this data. The NMR was done at ITQB with Luis. The team at SRF for the X-ray fluorescence image has been amazing over the years, supporting our projects on this bacteria. A special thanks to the amazing team of Impact that was fundamental not only for this project, but to really start up the cryo-EM in Portugal, and Instruct that has been for years and years has been really supporting all these researchers. And for those that work have not access to different methodology, Instruct plays a fundamental work. So thank you very much for giving the opportunity for access proposals in all the other activities that you organize. So thank you a lot for your attention.
Thank you, Celia. Please do put your questions in the chat. I will going to start off, Celia. I wanted to ask you, I mean, this is a loaded question, but the different kinds of granules, they have very, very different textures and shapes, and yet they have, from what you can see with the cryo EDX, the same elemental composition. Do you have speculation on what are the differences, and also if any one of them is including the nucleic acid content of the cell, the DNA?
Yeah, I mean, in one of them, the more amorphous, and the one I'm showing in the video, the one that is sharing share between the two cells. It's quite amazing because we can't see the DNA surrounding. So I don't know if this is like different stage of the granules because like when cells are dividing, maybe the way it's more easy if they are not very compact and then can go with the DNA. But I mean, based on the elemental compositions, yeah, right, they are the same. So we are trying to figure out what's the difference, but I'm sure I don't know yet the answer, but they are very amazing.
We have a question from Doriana Lamba. Is there any radiation effect following the EDX, and what about the metal oxidation state?
Well, the cells are vitrified, so I guess they are kind of more protected in these conditions. In terms of, so I would assume there's no radiation, or if there is really low. So the experiment that we did at SRF, the cells was not frozen, was not vitrified, so they were just dry out. So that's why we could use, they were like live cells, so we could use that to see the dislocalization of elements. In the EDX, I don't think it happens. I think they are maintaining.
Actually, they are, because in the tomography, we make sure to use low amounts of electrons so that we can get all the way through the tomographic tilt series. But with the EDX, we really blast them with electrons. So it's the electrons that are causing the damage, and the EDX signals coming out from there. But indeed, we're blasting as much as we can so that we can get high signal. So they are damaged. That's true. That is so.
Okay.
There's Mohammed Taha that says, thank you for this exciting talk. I've sent you an email with some questions, but my question now is, from where can one get an authenticated strain of the radiodurans?
I buy my strains to DSDM. It's a company in Germany that sells all these bacterial strains, and they are so I buy all my strains from there, and they are certified.
Okay. And then I had, okay. And so can you talk a bit more about the cooperativity between these different parts in the tetrad, and where you think, do you have speculation about the mechanism of the cooperativity?
Well, this is quite new for us. We know that bacterials, they share like DNA, but in terms of metal content, we didn't know. So this and what we see is that in fact the cells grown in M53, they are more resistant to radiation, UVC, and to oxidative stress. So I think they work like as a team, like working together. And I think it's much easier for them. It's like if one is damaged, then they communicate to each other, and then they kind of resist it in a more. So I think we are on a different level of organization. We are not only looking to a cell, we are looking to a multicellular formation and how they cooperate with each other. So this is really amazing.
One last question: is the metal distribution dependent on the cell cycle?
I don't know. We didn't went to there. We tend to use the cells always in the same phase. But I think we already tried in a different phase, but I think they were the same. I'm not sure, but we didn't went very deep in this part, but maybe.
Good. Thank you so much. Thank you both to Stefano and to Celia for these two beautiful talks, very different and both very interesting. And thank you so much to Instruct for organizing these wonderful meetings. And thank you for everyone for listening and participating.
Thank you. Bye-bye, everybody.
Bye, everyone.
Bye.
Thank you, Sharon.